Right, so now in this series of lectures, and we'll have about 3 or 4 of these, where we want to talk about molecular genetics. We're doing this because in order to understand Mendelian genetics, we have to kind of refresh our memory about what DNA looks like, a little bit about what chromosomes look like, and then most importantly, relative to Mendel, is understanding replication. And of course, we can't really talk about replication without talking about molecular genetics and how that starts out. So let's start from there. I want to bring forward, as we've been doing, we had a history lecture, and I want to continue to talk a little bit and include history and use history as the basis for sort of understanding where we go and how we got there. All right. So remember, we gave a definition of genetics, one that I like and kind of very common, is it's the branch of biology, the branch of the life sciences that deals with heredity, this how we transmit from one generation to the next, as well as, very importantly, the expression of inherited traits. How do we express those inherited traits? And that's the part where molecular comes in. Now, before we can really talk about the mechanism of expression, we have to understand the basis units that exist there to be expressed, and that's what we're going to do in these lectures. Okay? Now, we are going to basically talk about structure for now, structure of the genetic material. Later on, we'll talk more about the organization and the complexity of it, and those will come back when we return to this room, as I've called it, the molecular room. But for right now, we're going to talk more about structure. And what we're really interested in is getting to where we can start to understand how we get from genotype to phenotype. What is the mechanism that makes this happen? Now, we're going to do this throughout this whole process really by a fairly simple model. And that model is we look at variations or alterations that occur at the genotype and look how those affect the phenotype. You'll do that in the fly labs. You'll look at things, mutations. We'll look at allelic changes and see what effects they have. And even today, in the most sophisticated genetics labs, a lot of times what we do is we modify something in a cell, and we look to see what effects it has. We'll affect genotype, and we'll affect and see what kind of phenotypic effect. So that's really the model, and that's where a lot of this comes from. Now, in the beginning, there wasn't these existing animal or plant models to use, so most people tended to work with natural products. They tended to work with blood, or they tended to work with plant material or whatever, and they simply, as we talked about in the history lecture, there was just sort of these variations that would show up, right? These phenotypic differences, and he would kind of use those. Well, that's where everything started, and that's kind of where the story of DNA starts. All right, so think about genetics as a science, okay? Just to give you some real broad view of the history of this. We'll look at it. The information content, how much information is out there about genetics has shown just an amazing increase. We have to go back, if we're going to talk really about genetics, into the 1800s with Mendel and people before that. And then we move into that period between the late 1800s, middle 1800s, and the start of the 1900s. Okay, and that's period, particularly the period of the early 1900s, when there was the rediscovery of Mendel and all these new models that we could use for understanding DNA, as well as biochemical models and things came about. It's known as the classic period of genetics. And this is the area where we formulated some of the base theories that it took many years, in fact, sometimes quite a few decades, in order to prove or disprove those particular theories. Okay? Now, during this time, there was a pretty substantial growth in the information content. More and more publications were out there. We started to see a lot. But it really wasn't until about the 1940s that we saw an explosion of this information. From the 1940s into the 1960s, it was what is known as the biological revolution. And this is a period of time when all of biology, not just genetics, but almost all of biology had an explosion of information and forms. It could be in evolution, it could be in physiology and biochemistry and genetics and anatomy. All these areas, there was just a huge amount of information explosion out there. The 1800s and things had been kind of the primary center for things like physiology and anatomy. By the time we got into the 1940s, we then started to moving into genetics and into understanding these other areas of the biotic world. Now, since that time, into 2000s, where we are now, it's gone even more advanced, okay? We see, instead of having sort of a simple logarithmic change, we're seeing an exponential change. In fact, right now, the doubling time on information. In other words, how long does it take for the amount of information on a subject to double, to increase in the published world? That time is about 3 years right now in genetics. So from the time you enter college to the time you graduate, there'll be twice as much information known about genetic subjects. That's just phenomenal. There are just hundreds, if not thousands, of journals out there with thousands of articles being written and published on a daily basis. It is just an amazing time. Now, the question is, how did we get here, okay? How do we go from where we are with this massive amount of information coming along and these phenomenal techniques that are available and things that are happening now, way back to the beginning of our understanding? So we're going to have to walk our way back until the 1800s and then progress our way up until more recent times, okay? So what research contributed to defining the genetic material? Well, we're going to concentrate on 4 major areas, 4 major experimental groups. The first one started in 1927 with Griffith, and then that experiment, it was the basis for a series of experiments in 1994 by Avery, McLeod, McCarty. We'll talk about those as a group. We will look at these in the sense of them being the transforming principles. Okay? Transforming principles, it was what they were talking about, or transforming properties. These were heritable properties were carried as DNA, and that was the outcome of this series of experiments. We then will talk about, and this will be the last part of this series of lectures when we talk about them, we'll talk about Hershey-Chase experiments, which occurred in 1952, or at least were published in 1952, and these really set the basis for DNA is the biomolecule of heredity. Now, believe it or not, until 1952, there was still a huge amount of controversy about whether or not DNA was actually the unit of heredity. Nowadays, we all know it. We've known it from the time you were children. You've seen these things. Here's DNA molecule. Here's this viral. This is the biomolecule. But until middle of last century, there was great amount of argument about where it was. And in fact, it took the Hershey-Chase experiment to really nail it finally down. We'll also then, in the lectures that follow this series, we'll start talking about specifics of the genetic material itself. And a couple of primary contributors to that were Edwin Chargaff and Chargaff's rules about the composition of DNA and the chemistry of DNA. And then finally, the Watson-Crick models that we employ today in our understanding of what DNA looks like, the physical properties of DNA from Watson and Crick. Okay? All right, so it doesn't matter what the genetic material is. You have these questions about what is the chemical nature of this DNA, right, or this material, and we now know it's DNA. But it doesn't matter. It could be sulfur-based. It could be from anywhere, right? You have to think about what makes up genetic material. Well, the first question you ask before you even start digging into the physical part of it is, well, whatever it's made of, whatever materials, carbons, nitrogens, hydrogens, whatever's there, what does it take to be a successful genetic material? And in order to do that, we have to think about what elements would there be? Well, I think there are 4 elements, and most textbooks would agree there may list 3. Most of them tend to list 4. And so those are first that whatever the genetic material is, it needs to be able to store complex information very effectively. Okay? So we need to have a way to take a lot of information. I mean, there's a huge amount of information to build a sequoia or to build you or whatever that has to be held, used, and provided out to the phenotype. Okay? Now, because of reproduction, because we want to put that information into the next generation, it has to be able to replicate faithfully. We call this high fidelity. When I was a youth, we had hi-fis. That was the record players that were based on high fidelity records, right? Well, replicate faithfully. It needs to be able to take a nice copy of itself, not make many mistakes. If it does make mistakes, have maybe error checking or some other way like your computer does when you type into it, and then be able to put that out to the next generation. So replicate faithfully, however it's made. Okay? Obviously, it needs to have an effective way to encode phenotype. So not only does it need to store information, but somehow there has to be a way to get that information off of there and out to a way that can be seen or used, such as a phenotype. So it has to be capable of that. And then finally, it's really important that genetic material be mutable. The world around us is constantly changing. The environment changes, the food sources changes, our interactions change. And generation over generation, we have to have a way for our genetic base, our genome, to actually change as well. So it has to be mutable. It has to be some way that we can get some new variation out there. Otherwise, we're static, and things that are purely static tend to die out. They don't tend to persist. Okay, so these are the things that we need when we have this thing. Now, how are we going to get there, right? Well, let's start back. Let's go back into the 1800s and say, what did we know? What did we know about DNA or about our genomic material back then? Well, surprisingly, we knew quite a bit, okay? As early as 1869, so shortly after the Civil War in our country, in Switzerland, there had already been the discovery and, in fact, the isolation of what we now know to be DNA. Frederick Mincer had, working in, as you can see in this picture here, working in this old castle, literally in the old kitchen and under deplorable conditions, began to isolate nuclein, what he called nuclein. He called it that because he believed it came from the nucleus of the materials that he was working with. Now, what he was initially interested in was isolating material from white blood cells. In fact, it was a little bit gross, but what he was using was bandages from the hospitals that had this pulsy white material, which turned out to be white blood cells, that he was isolating the first DNA from. And this little vessel picture that you can see here, this little test tube, is actually DNA that still exists that he isolated later on from salmon sperm, but it was still DNA. So he worked through a whole series of complications to get DNA isolated. But even in the 1800s, he knew that this stuff existed, and he knew it was called nuclein. Okay? Now, from that, it was actually one of he was a student of Aldrich Kinsler, and Kinsler went on to take these ideas and to actually pull apart this stuff called nuclein. Starting in the 1870s and so, he experimented on this nuclear material after Mincer had discovered it. He developed ways to more effectively isolate it, and eventually he was able to realize that that the material was nucleic acid. Working after that, I've got 1885 on up to about 1901. During that period, he was actually able to isolate and recognize that this nuclear material, which he was accredited as calling DNA because it was a deoxyribonucleic acid, he realized there are 5